Today I am talking to you with a very important topic, that is to testing the genetic purity of a variety by the means of electrophoresis. In the previous lecture, I am also discussed to test the genetic purity of a variety or a seed by means of grow out test. So grow out test is used to testing the genetic purity of a variety on the basis of their morphological characters, or this grow out test is conducted in the field conditions. But in case of electrophoresis method, we have to be conducted the experiment in the laboratory condition, and we have to be find out the true type seeds with the help of molecular biology techniques, that is the electrophoresis method. So how to test the genetic purity of a variety? By this biotechnological methods, molecular biology techniques methods, that is the electrophoresis method. Electrophoresis is a technique, it is a biotechnological tools, biotechnological techniques in which we have to be separate the charged biomolecules on the basis of electric current. Electrophoresis is a set in which we have to be find out the differentiate the different types of the biomolecules on the basis of their molecular weight by means of the providing the electric current. So we have to be separate the biomolecules such that the DNA molecules, RNA molecules as well as proteins. So what is the procedure to checking the genetic purity of a variety? The procedure is that in case of electrophoresis method, electrophoresis, in electrophoresis we have to be firstly prepare the gel, and that gel is prepared on the basis of your biomolecules. Suppose you have to be isolated the DNA from your plant sample, so you should prepare the agarose gel, but you should have to be isolate the protein from your variety, from your plant, from your seed. You have to be prepare the acrylamide gel, polyacrylamide gel. For separation of the protein, you have to be prepare the polyacrylamide gel electrophoresis. And for the DNA separation as well as RNA separation, you have to be prepare the agarose gel. So this is the two types of gels are prepared to find out the true type of variety, or we have to be separating the different types of biomolecules by help of the two types of gel, agarose gel as well as polyacrylamide gel electrophoresis. PAGE, the full form of PAGE is polyacrylamide gel electrophoresis. The molecular weight of the DNA is more as compared to the protein, so that the DNA is separated by agarose gel, because the pore size of the agarose gel is more as compared to the polyacrylamide gel. The pore size of polyacrylamide gel is smaller than the agarose gel. So how to be prepare the agarose gel? So today I am discussing to testing the genetic purity of a variety by the isolation of the DNA molecules from your plant sample. So for the DNA isolation and checking the quality on the basis of the DNA isolation, you have to be prepare the agarose gel. For preparation of the agarose gel, you have to be use the agarose powder in 0.8 to 1%. The agarose solution is made in 0.8 to 2% solution is prepared by using the two types of buffer. TAE buffer as well as TBE buffer. This buffer constant the pH of the solution. TBE buffer, tris butyrate EDTA buffer, or TAE buffer, tris acetate EDTA buffer. So normally two types of buffers are used to making the agarose gel. So here we have to use the TBE buffer, 0.8 to 2% solution of the agarose, and we have to use the conical flask. We measure the agarose powder, we put it into the conical flask and make up the volume, and then boil this agarose powder, putting in the TBE buffer, and boiling at that duration when the clear cut transparent solution is appear. When the transparent solution is appear, you have to be take from the oven and added in this conical flask the one important intercalating agent, that is the ETBr, ethidium bromide. So why we adding the ethidium bromide, ETBr, in the solution of the agarose buffer? Because of, if we are not adding the ETBr to making the agarose gel, we cannot visualize the DNA molecules in UV ray. This ETBr is fixed with the nitrogenous base of the DNA molecules. The adenine is paired with the thymine and guanine is paired with the cytosine, and the both bases are attached by the hydrogen bond, and this intercalating agent is also attached to this nitrogenous base. And after visualizing this DNA molecule in the UV ray, you have to be observed in a light emitted from these molecules, and we can easily visualize the DNA particles. Because of that reason, we have to use the ETBr in that solution. And after adding the ETBr in the agarose gel, we can carefully pour this agarose gel solution into the gel casting tray. And after the pouring of the agarose buffer in gel casting tray, after the 30 minutes when the gel is solidified, you have to take and put into the filled TBE buffer of the whole setup of the gel electrophoresis. And in the well mentioned in the gel, the first lane of the gel, you have to be loaded the ladder. Ladder is nothing but it is a molecular scale. It is the different fragment of the DNA where the molecular weight is known, and on the basis of the ladder sequence, on the basis of the molecular weight of the ladder DNA, we have to find out our DNA sample size. According to the molecular weight of the ladder, we have to find out the molecular weight of our test sample. So the first lane you have to loaded the ladder DNA. The next lane you have to added the different types of test sample. The test sample is those sample where we have to isolated the DNA for the checking of the genetic purity. In the case of testing the genetic purity by means of electrophoresis, you have to first of all isolated the DNA from the different types of plant samples by the means of CTAB method. CTAB method is used to isolating the DNA from the plant. Cetyl trimethyl ammonium bromide. The full form of CTAB is the cetyl trimethyl ammonium bromide. So this is a very important molecular biology techniques. By the help of electrophoresis method, we have to easily separate the different types of the test sample. Suppose the 5 plants where the genetic purity is to be tested, and in 5 plants the 1 or 2 plants where the doubted sample is here. So how to be clearly differentiate between the original sample with the doubt sample? So first of all, we have to be isolated the DNA from the your samples, your doubted samples, as well as original sample. Original sample means authentic sample. The authentic sample is the breeder seed as well as nucleus seed, where the clear cut information has to be mentioned in the original plant breeder. That sample is also used here because of the comparison for the test sample, like the doubted sample. So after the isolating of the DNA from the different plant samples as well as standard samples, you have to be digest the DNA sample with the restriction endonuclease enzyme. Why we have to be digest your DNA sample with the restriction endonuclease enzyme? Because this restriction endonuclease enzyme, it is a type of the molecular seizure. It is the capacity to break down the DNA fragments, break down the DNA samples into different fragments and cutting from the special types of sequence. It is not random cut is produced from the DNA sample. This restriction endonuclease enzymes is to cut the specific location in the DNA samples. It cannot cut the random point of the DNA. The specific recognition sequence. And that sequence of the DNA where restriction endonuclease enzyme is cut down, that sequence is known as recognition sequence or restriction site cutting sequence. Suppose there are several types of restriction endonuclease enzyme, like the ECO R1, HIND second, HIND third, BAM H1. There are lots of restriction endonuclease enzymes are used in molecular biology to doing the several research works. So after digestion of the DNA samples from the restriction endonuclease enzyme, suppose your authentic sample is breakdown from the one location. So 2 fragments are generated in a standard sample or authentic sample. So the other 5 plants is also digestion with your restriction, same types of restriction endonuclease enzyme, not changing the restriction endonuclease enzyme. The same restriction endonuclease enzymes is used in the authentic sample as well as your test samples. So in case of the original samples, there are 2 fragments is observed. According to that sample, the standard sample, we have to be compare from that sample. The 2 fragments, the 2 bands are seen in the gel electrophoresis by visualizing in the UV ray. The 2 fragments are seen in a standard sample. According to that sample, we have to be compare your test sample, and those test sample, the same types of bands are appear as compared to your standard sample. That sample is the original. Suppose the plant number 1 produce the 2 bands like the standard sample. So this is the genuine type of variety. The plant number 2 is also produce the 2 types of fragment. Means 2 bands are appear. This is also the true type of variety. But in case of plant number 3, 4 or 5, the fragments is the different. The 4 fragments, the 1 fragments, not the 2 fragments. So this is the mixture of the variety. This is not a true type of variety. So according to the number of the band, number of the fragment, we have to be easily differentiate between the test sample and a standard sample and comparing with the standard sample. So this is the type of the genotyping according to the number of the bands appear in the gel. The important point is also mentioned here that the DNA is negative charge. So DNA is loaded in the negative charge of the gel electrophoresis. So the movement is towards the positive charge. The smaller size fragment is move fast, and where molecular weight is more is slow run in the gel electrophoresis. So according to on the basis of the ladder sequence, we have to easily find out the molecular weight of our test sample, and according to your standard sample, the number of fragment you have to be compare with your test sample and easily find out the genetic purity of your variety. If the fragment size or number of fragment is same with your standard sample or authentic sample, your genetic purity of variety is true. So this is the overview of the testing the genetic purity by means of the electrophoresis. So this electrophoresis is not using to separate the only DNA molecules. DNA molecules is separate by preparing the agarose gel, but you have to be isolating the protein molecules. You have to be prepare the polyacrylamide gel, the SDS phase, sodium dodecyl sulfate. So in this lecture, I am talking about the very important information regarding the genetic purity of a variety to testing by means of the gel electrophoresis and separating the different types of DNA fragments, different types of DNA molecules of the different variety, and you can easily differentiate of the lots of variety in a very less time in the laboratory condition. This is a in the very short period of time you have to be calculate the data according to the genotypic data. The lots of molecular markers are also used to testing the genetic purity of a variety, but this method is used the non-PCR based marker, that is using the restriction enzyme. So this type of procedure is known as restriction fragment length polymorphism. This is also a type of molecular marker, but this is a hybridization based marker. This is a non-PCR based marker. This is a codominant marker. Codominant marker means the codominant marker is those marker by using this codominant marker we can easily differentiate between the homozygote as well as heterozygote. So this is the special feature of that RFLP marker, restriction fragment length polymorphism. Okay, thank you.